this video, we are going to cover the structure of prokaryotic cells and viruses. In previous videos, we have covered the structure of eukaryotic cells, but in this video, we are going to cover the structure of prokaryotic cells. Now, they do share some of the same features, but we're going to explore the more specific features that are found in a prokaryotic cell. Most prokaryotic cells are bacterial cells, and they're relatively successful due to their small size. They are around 0.1 to 10 micrometers in length, and they are much smaller than the eukaryotic cells that we were looking at in the previous video. They're also much simpler than eukaryotic cells. They contain no membrane-bound organelles, so no nucleus, no mitochondria, or anything like that. They do, however, have a cell membrane around the outside. So in the same way that your eukaryotic cells have. Okay, so this still works as an entry and exit system, still contains your phospholipids that have your fatty acid tails and your phosphate head. And like I said, it's still there to control entry and exit of substances into and out of the cell. Our prokaryotic cells don't contain any membrane-bound organelles, but they do contain ribosomes. Now, they have ribosomes in the same way eukaryotic cells do, but they are smaller. So they are called 70S ribosomes instead of 80S ribosomes. They still have two subunits, and they are still involved in protein synthesis. Okay, so they have the same function. They're made of rRNA and proteins, exactly the same as your eukaryotic ones, but they are just free-floating in the cytoplasm. Okay, they're not connected to any endoplasmic reticulum like they are in the eukaryotic cells, because bacterial cells don't have those membrane-bound organelles, so therefore don't have any endoplasmic reticulum. Okay, so ribosomes still present, they are smaller at 70S, they're still made of rRNA and protein, and they still do protein synthesis, but this time they are only found in the cytoplasm rather than attached to any endoplasmic reticulum. So because our prokaryotic cells have no membrane-bound organelles, it follows that they have no nucleus. Their genetic material instead is simply free-floating in the cytoplasm. So let's zoom in and have a look. Here is our bacterial DNA. Now it's in the form of one long circular strand of DNA, and it's not associated with histones. Now, histones are proteins that you find associated with the DNA in eukaryotic cells, and you will study that when we look at the structure of DNA in eukaryotic cells more specifically. Now, they also have additional DNA in the form of plasmids. These are usually represented as these circular diagrams when they are drawn. And these provide the bacteria with additional kind of needs, such as antibiotic resistance. Now, the really clever thing about plasmids is they can reproduce independently, so the bacteria can make more and more of them. They can also share them. So really cleverly, they can share these plasmids between each other. So if there is antibiotic resistance on this plasmid here, a bacteria can share it so that antibiotic resistance can kind of spread between a bacterial population, which I guess is good for them, but not so good for us. So prokaryotic cells then have this circular loop of DNA. It's not associated with histones, and they have these additional sections called plasmids. Prokaryotic cells also have a cell wall, just like some eukaryotes, but it's important to mention that our plant cells have a cellulose cell wall and our fungi have a chitin cell wall, but prokaryotic cell walls are made of murin, which is a glycoprotein, so it's different. So when you're thinking about exam questions, it's important not to simply write cell wall, but to be specific and mention that it's made from murin. Prokaryotic cells also have some additional features that we need to know about: flagellum, slime capsules, and pili. So let's have a look at flagellum first. These are projections on the outside of your bacteria cell, and they help to propel your bacteria cells around. They have this rotation movement as opposed to a flapping motion to help with the movement. Now, in this case, there's just one flagellum. If there were multiple, then they would be called flagella. Slime capsule, that's on the outside of your bacteria cell, and it's there for protection. Now, usually pathogenic bacteria have these because they require the extra protection. And finally, we have pili. These are hair-like structures on the outer surface of your bacteria cell. They can be used for adherence to surfaces or to each other, and in the transfer of genetic material. They can be localized or they can be spread all the way around. Now, it's important to note that these features are not always present in your prokaryotic cells. So just think about the exam questions carefully and check what they are actually asking you. So these are not always going to be present when you're comparing to eukaryotes, for example. So just check really carefully. Sometimes in exam questions, they like you to do comparisons. So let's have a look at a typical kind of comparison. They would ask you about prokaryotes, but not eukaryotes. So some things that are found only in prokaryotes, but not eukaryotes, would be circular DNA free in the cytoplasm, murin cell wall, important that you don't talk about just a cell wall because obviously we have cellulose and chitin found in plants and fungi, and 70S ribosomes, again not just ribosomes because obviously eukaryotes have them as well. So exam tip, answers that would not be acceptable. So flagellum, slime capsules, not always found in your prokaryotes. Chromosomes, because they would be found in your eukaryotes. And plasmids, because again, sometimes these aren't found in your prokaryotic cells. So things that are definitely always found, okay, so your circular DNA, your murin cell wall, and your 70S ribosomes freely in the cytoplasm. Another really type of common exam question that they ask you to do is to compare the structure of the DNA specifically within your prokaryotic and eukaryotic cells. So let's have a look at that. Now, I know that you haven't done the DNA structure of the eukaryotic cells yet, but don't panic. We're putting it in now so that we can clearly make that comparison, and you will come back to it later in more detail. So prokaryotes versus eukaryotes. Firstly, free in the cytoplasm, okay? So that DNA is not contained within a nucleus like it is within the eukaryotic cell. Next, no histones. So these are the proteins that help the DNA in the eukaryotic cells coil up, but not present in your prokaryotes. There are plasmids in prokaryotic cells, but none in your eukaryotic cells. There are no introns in your prokaryotic cells either. Now we do have introns in our eukaryotic cells, and again you will come back to what introns are when we do DNA in your eukaryotic cells. DNA in prokaryotic cells is shorter, okay, much shorter than the longer DNA that you find in your eukaryotic cells. And finally, circular DNA as opposed to linear DNA. Okay, so we were talking earlier that your circular DNA in your prokaryotic cells is kind of one long big loop, okay, whereas it's linear in your eukaryotic cells. Okay, so this is an exam tip. And again, if you're answering this in a question, you can write it in a table. It really helps you make those direct comparisons that's needed in order to get the marks from the question. Having looked at the prokaryotic cell structure, we are now going to move on and have a brief look at viral structure. Now, viruses are acellular and non-living, okay? So that means that they don't follow those usual life processes that living cells do, okay? Some examples are HIV, that causes AIDS, influenza, like flu, and rhinovirus that can cause the common cold. Now, the difference between these and your prokaryotic cells is that they need a host cell in order to replicate. And we will look at viral replication later in the course. You need to know about some specific parts of your virus, and the main parts are a capsid, attachment proteins, and genetic material. So capsid, in more detail, is this outer layer of your virus, and it's there really just to protect that inner genetic material. Your attachment proteins are on the outside, and these are the parts that allow them to attach to their host cell, also where the antigens are found. And finally, the genetic material on the inside. This is what's injected into the host cell in order for the virus to replicate, and it can be both RNA or DNA depending on the virus. Now, you will look at the structure of the HIV virus in more detail later in the course and also have a look at how those viruses replicate. But for now, you just need these basic structural points, so the capsid, the attachment proteins, and the genetic material. In this video, we covered the structure of prokaryotic cells, we compared prokaryotic cells with eukaryotic cells, and we looked at the structure of viruses. The next set of videos are going to cover the methods of studying cells.